Hello students, in this lecture we'll be going through something called synaptic transmission, right? So we know what is synapse. Let's say this is the presynaptic neuron and let's say this is the postsynaptic neuron. So we can say that this is presynaptic neuron and this is postsynaptic neuron. So the junction between this presynaptic neuron and postsynaptic neuron, there is a cleft, or we can say there is a gap, and this gap is known as synaptic cleft. Okay. Now, in this part, in this presynaptic neuron, there are several vesicles like this, and these vesicles have different types of substances that we call neurotransmitters. These are chemical substances. Okay, and in this case we also have some channels like this. All right, and in these, this is the extracellular fluid, right? Extracellular part, or that means outside the neuron, and this is the inside of neuron. Normally what happened is the sodium and calcium ion concentration remain very high in this extracellular part. Okay, when any action potential comes, let's say this is the action potential or the impulse. We can say this is the impulse. When it comes, these calcium channels, these are calcium channels. These calcium channels are opened up. They get opened. So what happened? Let's say these are also the calcium channel, and this calcium channel is now opened. As a result, the calcium ion will enter. So this calcium ion enter into this presynaptic neuron. So this calcium ion is also entering. Let's say there is also calcium ion, and this calcium ion enters. When this calcium ion enters, a reaction happens between these vesicles and this membrane. So these are actually vesicles, right? And these green colored things, these are neurotransmitters. All right. So when this calcium enter, something happen, and as a result, these vesicles get attached to this membrane like this. Sorry, this vesicle get attached with this membrane like this, and the neurotransmitters get released in here. Okay, but the thing is, what happened in this area? What happened in this area? We'll discuss over here. So let's say this is the vesicle, and these are the neurotransmitter, and this is the our presynaptic membrane. This is the presynaptic membrane. All right. In this part, we have some proteins. We call them the V snare protein. And in the presynaptic membrane also, we have some other proteins, and we call them the T snare protein. the T snare protein. V is for vesicle, okay, and T is for target, because the target of this vesicle is the presynaptic membrane. This is the presynaptic membrane, right? Presynaptic membrane. This is like this, okay? So when this V snare and T snare, they get attached to each other, there is a process called vesicle fusion happen. So when V snare and T snare are interacting, there is a process that happen, and that is called vesicle fusion. That means this vesicle is now fused with the presynaptic membrane, and all the neurotransmitter, they get released out, okay? So we can also call this process as exocytosis. All right, so this exocytosis happen in here, and the neurotransmitter get released. Now there are some receptors in here. There are some receptors in here with which these neurotransmitters can bind. And when these neurotransmitters get bind, these are the neurotransmitter receptors. When these neurotransmitters get bind with this receptor, some channels are again opened in here. Okay, let's say these are the channels, and these are basically sodium channels. So these are sodium channels. Now, as I have already mentioned that in the extracellular fluid, both calcium as well as sodium was in higher concentration. So calcium was also in higher concentration, also sodium was in higher concentration. Now this sodium will enter through this channel. So sodium enter through this channel. And when sodium enter through this channel, what happened is depolarization. And due to this depolarization, this postsynaptic neuron become excited. Okay, and then maybe action potential will be generated. Now to produce this action potential in this postsynaptic membrane, we need these neurotransmitters to be excitatory. Okay, so like we can say that neurotransmitters can be of two types. One is excitatory and another one is inhibitory. Like if we have acetylcholine or ACH, that's an excitatory neurotransmitter for skeletal muscles. And if we have GABA, that's an inhibitory neurotransmitter. So let's say this neurotransmitter was acetylcholine and that is excitatory. So action potential will be generated in this part, okay? And then through this, the stimulus or the impulse will be propagated from one neuron to another neuron. Okay, so this is the overall mechanism of synaptic transmission. This exocytosis of vesicles with the presynaptic membrane and binding of this neurotransmitter with this neurotransmitter receptors, and again the formation or generation of action potential in the postsynaptic neuron. This whole thing is called together as synaptic transmission. Okay, I hope you have understood this thing, and if you have any queries or question, please let me know. Thank you.